Another important test is this variance mean dependence. And this is basically looking at what is the distribution of variance at different levels of fluorescence intensity. As fluorescence intensity increases, sometimes the variance increases along with it. And if it does, that means you're going to bias your analysis to seeing a lot of variance in highly expressed genes and probably missing some of the lower expressed ones. So by looking at the variance of mean dependence, the red dotted line, it should be fairly flat throughout the range of expression. In addition to looking at the housekeeping genes, GAPDH and beta-actin for RNA quality, one can also look at overall RNA degradation plots. In many of these chips, there's approximately 11 probes per gene. And while an individual gene may have different intensities across these chips, there is a tendency, just like with the beta-actin and the GAPDH, to have higher expression at the 3' end than at the 5' end. So when all of the most 3' probes are averaged and ranked down to the most 5', you should get a slope that is indicative of the RNA quality. What's important to look at is the value of the slope itself, but probably more important is are all the chips similar in their slope. In this case, they do all look fairly similar. Another plot that is sometimes useful in looking for it is looking for relative log expression, and this is looking at the relative expression of each individual gene compared to the median expression for that gene across all chips. And this should be fairly uniform and set around 0. What we can see here is our first clear clue that chip number 1 may be an outlier and not just chips 6 and 8, which we saw earlier, because the relative log expression is much wider for this chip than the other chips. Going a step beyond that is this so-called normalized unscaled standard error plot, which, similar to the previous plot, takes the standard error for each of on the expression for each of these genes compared to the median, and looks at that across chips. Now, in this, the median should be centered around 1, and obviously a small error around that. And again, chip number 1 in this case has a much higher relative unscaled standard error that suggests again that it also may be an outlier. In addition to the other tests that we do at Precision Biomarker Resources, we also provide a summary of the Affymetrix recommended QC stats, including the GAPDH, actin 3', 5' ratios, the kind of the scaling factor, which is in the blue band, the bars, and the percent present calls and the background. Percent present calls are in the left-hand column near the cell file name, and that's for example, in 1753.cell, that would be 60.25% in blue, and the number below in red gives basically the background calculation. Since all the values in the QC stats part of this graph are in blue, it indicates that they're all in bounds via Affymetrix QC stats, which are all very good, but you can see by looking at some of the between chip comparisons and that some of those are much more sensitive to detection of outlier than the basic Affy QC stats. We also provide sort of separation of the perfect match and mismatches, looking at their overall intensities. Now, this data was then normalized using RMA. And again, if we look at the summary, what we see in this summary is that after RMA normalization, chips number 6 and number 8 are now within bounds. But it's this chip number 1, which was identified particularly in the normalized unscaled standard error plot, that is still out of bounds. And after normalization, its M versus A plot, box plot, and heat map all suggest that it's an outlier array. So if we go back and we look at this, remember we said that the blue or the density lines for these M versus A plots should be straight all the way across and fairly narrow, and we can see for most of the chips that's the case, except for chip number 1, it's still kind of broad. If we go on and look at the array intensities, either in the histogram or the box plot, we see that the distribution of fluorescence intensities is a little narrower in number 1, so providing a little higher peak. On the histogram, it's denoted by the dashed line as opposed to a solid line, and on the box plot, it's in gray. The rest of the chips normalized very well with RMA. On the box plot, the heat map, you can see that it's still an outlier. And on the principal components analysis, it's the one that's down here in the corner and away from the rest of the chips. Now, why is this outlier detection so important? If we go back to this experiment and think about the experiment itself, we're looking at cerebellar gene expression from 5 wild-type mice, 15 weeks of age, and 5 ataxin 1 knockout mice, cerebellar gene expression, 15 weeks of age. This ataxin 1 is a protein of unknown function associated with cerebellar neurodegeneration in spinocerebellar ataxia type 1, which impairs movement of the eye as the mice develop. Now, when we go through quality assessment and look at all 10 of these chips, and then perform a moderated t-test to look for differentially expressed genes, at a p-value level of 0.01, there's only 34 genes that we find differentially expressed. When this is further corrected for a false discovery rate, and now we look at genes that are 0.001, there are only 4 genes. Now, taking this on to go beyond to a pathway analysis, these 4, based on these 4 genes, a neuroactive ligand receptor interaction pathway was identified in KEGG with a corrected T value of 5.65. However, if we take out array 1, which we identified as an outlier, you can now see that instead of 34 genes at a p-value of 0.01 or less, there are now 190. And there are 14 genes at this p-value of 0.001. Now, if we take this on and look at the most enriched KEGG pathway through pathway analysis, that now we're able to, without array 1, identify 23 genes in this pathway, giving it a much more significant corrected T value. And that's the point of improving your confidence in this kind of data. So was taking out array 1 a specific effect, or was it just using less data? So if we go through and sequentially take out each chip and look at the column p-value 0.001, there's not much difference when you take out any other chip. It's only when you take out chip 1 that you get a significant increase in the number of genes that are still surviving a false discovery rate analysis. So in summary and conclusion, I hope I've given you some idea of why quality assessment is important. It's very much still needed. It really does give one a first taste of the data and suggest what might be done for subsequent analysis. And in some ways, I hope I've shown that by removing an outlier, that the statistical power of your experiment can actually be increased. So thank you very much for your attention, and I hope this was helpful in understanding the importance of quality assessment and the importance and understanding your quality assessment report from Precision Biomarker Resources. Thank you.